A mammal is defined by any animal of the Mammalian, a large class of warm-blooded vertebrates with a four-chambered heart. The generic mammalian cell has many organelles. Firstly, it is made out of the nucleus. The nucleus is the brain of a cell and is made up of deoxyribonucleic acid (DNA), nucleoplasm, nuclear matrix, and ribosomes. The DNA contains all the genetic information. It can be tightly or loosely packed depending on the activity of the gene expression. The nuclear envelope is the most important feature as it distinguishes between eukaryotes and prokaryotes. It is a double layer which serves to compartmentalize genetic material and is a barrier against cytoplasm ions and solutes. In the cell, there's also a mitochondrion. It has a crystal structure and is classified into inner and outer membrane. The matrix of the mitochondria possesses its own DNA, which is distinct and separated from nuclear DNA. It has meteorized that it was hijacked from the bacterial cell millions of years ago. The rough endoplasmic reticulum is attached to a cytosolic surface and have a rough appearance. It is involved in protein processing and serves as molecular chaperones to ensure proper protein folding. It is extensively found in pancreas and salivary glands. On the other hand, smooth endoplasmic reticulum (SER) does not have ribosomes. It is involved in the synthesis of hormones, detoxification, regulation of calcium. It is extensively found in muscle cells, kidney tubules, and endocrine cells. The Golgi complex is involved in post-translational modification. It assembles carbohydrate components and transport of proteins out of the cell via the cell surface. The lysosome is the cell digestive organelle and break down materials brought in from external environment. The peroxisomes are involved in fatty acid and hydrogen peroxide degradation. The function of endosomes is to sort and transport proteins from Golgi complex to lysosomes or vacuoli. The cytoskeleton is made up of a variety of related proteins which direct the movement of materials and organelles within the cell. The basic unit of life is the DNA. The DNA does not have ribose and is instead replaced by hydroxyl, therefore the name deoxyribonucleic acid. The nucleic acid is made up of a sugar phosphate backbone which consists of a nitrogenous base, a 5-carbon sugar, and a phosphate group. This forms nucleosides and nucleotides. A nucleoside is made up of base and sugar, while nucleotides are made up of base, sugar, and phosphate. There are two types of base, purine and pyrimidine. In DNA, adenine and guanine are purine base, while cytosine and thymine are pyrimidine base. In RNA, cytosine and uracil are pyrimidine base. Adenine binds with thymine while guanine binds with cytosine. This is known as complementary base pairing. Phosphodiester bond forms when they bind to each other. The bonds are covalently bonded. The reason why DNA coils is because it loops around histones forming the nucleosome. It then forms coils of nucleosome called chromatin. The chromatin then forms chromosome. Humans have 23 pairs of chromosomes. There's a conventional way of reading gene. 5' indicate that there's a 5-carbon sugar with a phosphate group, while the 3' indicates that there's a 3-carbon sugar with a hydroxyl group. This gives the gene a direction and also means that DNA is anti-parallel. 5' to 3' is known as antisense, while 3' to 5' is known as sense strand. DNA undergoes replication. Firstly, helicase unwinds the double strands of DNA. Secondly, the single-stranded binding protein binds to DNA strands. Thirdly, the primase creates primer on both strands. Primase is an initializer which tells polymerase where to start. Next, DNA polymerase begins to build. As mentioned previously, the two strands of DNA are anti-parallel. DNA polymerase can only work for 5' to 3' direction. This means that the 5' to 3' is the leading strand, while the 3' to 5' is the lagging strand. This results in Okazaki fragments, which are gaps. Therefore, ligase will have to close up the gaps. DNA replication is semi-conservative. This means that the two copies produced will each contain one of the original strand and one new strand. DNA replication is highly important to ensure survival of organism. It has to be highly accurate to reduce the risk of mutations.
In translation, the tRNA is activated. Specific initiator tRNA binds at start codon AUG at 5' end of mRNA with the aid of initiation factors and GTP. So mRNA is translated in 5' with 3' direction. Initiator tRNA then binds to mRNA in P site. This is known as initiation. In elongation, the second amino acid cell, tRNA enters site A. tRNA anticodon is complementary to mRNA codon in A site. A peptide bond forms between the two adjacent amino acids. The bond between amino acid and tRNA in P site cleaves. tRNA in A site has two amino acids known as peptidyl tRNA. Ribosomes move one codon along mRNA towards the 3' end. Uncharged tRNA then moves from P site to E site, after which it leaves the ribosome. Peptidyl tRNA then leaves ribosome and binds to P site. After translocation, amino acyl tRNA with the correct anticodon binds the newly exposed codon in A site. Termination is signaled by 3 stop codons. These stop codons do not code for any amino acids. The cell cycle is a series of events that take place leading to division and duplication of DNA. There are some differences between meiosis and mitosis. A few would be: firstly, the purpose of meiosis is to form gametes, while mitosis is for growth and repair of cells. Secondly, meiosis is a form of sexual reproduction, while mitosis is a form of asexual reproduction. Lastly, meiosis involves 2 rounds of cell division, while mitosis involves only 1 round. Meiosis 1 is known as reduction division, as the number of chromosomes are reduced from 46 to 23. This is also known as a separation of homologous. In prophase 1, there is a crossing over. This allows for exchange of genetic information, which results in genetic recombination. This is followed by the breaking down of the nuclear envelope. In metaphase 1, the centrioles are at opposite poles of the cell. They will then be aligned on a plane equidistant from the poles called metaphase plate. In anaphase 1, the two chromosomes separate and start moving towards opposite poles of the cell. The mitotic spindles are now assembled. A new nuclear envelope forms around each chromosome set. This is then followed by cytokinesis, which will result in 4 daughter cells known as haploid. Cytokinesis is where the cell will further split and divide. The second round of meiosis is similar to mitosis. It results in the separation of sister chromatids. In prophase 2, sister chromatids condense while the nuclear envelope disappears and the spindle apparatus forms. In metaphase 2, the 2 daughter cells produced by the first meiotic division moves to opposite poles of the cell and will be aligned along the metaphase plate. In anaphase 2, sister chromatids separate at the centromeres and move to opposite poles. In telophase 2, a new nuclear envelope forms around it. The mitotic spindle disassembles and the Golgi and ER reforms. This results in differences between chromosomes in meiosis and mitosis. In meiosis, there is a pairing of homologs and crossing of chromosomes occur, while in mitosis, there is no pairing and no crossing over of chromosomes. The end product of meiosis is 4 genetically different gametes, known as haploid cells. On the other hand, the end product of mitosis is 2 genetically identical daughter cells, known as diploid cells.
Apoptosis is self-death marked by a well-defined sequence of morphological changes. Self-death is necessary to control multicellular development, regulate number of neurons in the nervous system. It is also important as it deletes necessary structures such as the tail in developing human embryos. Furthermore, the majority of cells generated during development also die during development. There are different pathways and apoptosis mediators. This is different from necrosis, which is self-death due to tissue damage. DNA mutations are permanent alterations to the DNA nucleic acid sequence. They provide biological diversity by creating genetic variation when organisms evolve and adapt to environment changes. Hence, DNA mutations are the driving force for evolution. However, DNA mutations sometimes result in the increased susceptibility to disease and cause phenotype autoimmunity. One type of DNA mutation is point mutation, which is a mutation that affects only one or very few nucleotides in a sequence. Different forms of point mutation include insertion, deletion, and substitution. Insertion mutation is the addition of one or more nucleotide base pairs into a DNA sequence when deletion mutation is the opposite. It occurs when part of DNA molecule is not copied during DNA replication.
These two types of point mutation can drastically affect the sequence of amino acids produced. A substitution mutation occurs when one's base pair is substituted for another. Transition happens when a purine is changed to another purine or when a pyrimidine is changed to another pyrimidine. Transversion, on the other hand, happens when a purine is changed to another pyrimidine or vice versa. Insertion and deletion mutation leads to a frame shift mutation. When one or two bases are added or deleted, all of the 3 base codon change. This completely changes the amino acids that will be produced, which in turn changes the structure and function of the resulting protein and can render it useless. Substitution mutation leads to 3 kinds of mutation: missense, nonsense, and asylum mutation. Missense mutation occurs when one nucleotide is substituted and a different codon is formed. This results in the new codon producing a different amino acid sequence that is to be incorporated into the protein chain. A missense mutation is considered conservative if the amino acid formed via the mutation has similar properties to the one that was supposed to be formed instead. Similarly, nonsense mutation also occurs when one nucleotide is substituted. However, this time, a stop codon instead of a codon that codes for an amino acid is formed. It introduces a premature stop codon in the DNA sequence, resulting in a shorter, unfinished protein product, which is also known as a truncated protein. Silent mutation occurs when a nucleotide is substituted, but the same amino acid is produced anyway. This can occur because multiple codons can code for the same amino acid. This results in no change in the final amino acid sequence and thus no change in the final protein product. Gene mutations can be classified under 2 categories. Firstly, by origin and secondly, by cell type. Cell type classification refers to the mutation occurring either in somatic or germinal cells. Somatic cell mutation is perpetuated only in somatic cells and is replicated from original mutant cell, while germinal cell mutation occur in gametes and transmitted through generations. The origin of the mutation can either be induced, which occurs when a mutation reacts with the parent DNA, causing a structural change and affects base pairing capability of altered nucleotide, or spontaneous, which is caused by spontaneous errors in replication, resulting in DNA sequences replicated. This is because the mutagen is able to evade proofreading function of the DNA polymerase at the replication fork. Spontaneous mutation can be caused by base substitution mismatch during DNA replication, which in turn results in the formation of tautomers and small additions and deletions by DNA looping out errors if not corrected. Spontaneous chemical changes may cause structural alterations, which results in depyrination and deamination due to inherent chemical instability of N-glycosidic bond. On the contrary, induced mutation can be caused by radiation, base analogues, base modifying agents, and intercalating agents. DNA repair systems can be classified as either direct reversal repair or excision repair. Direct reversal repair includes mismatch repair by DNA polymerase proofreading,
repairing and repair of the UV-induced pyrimidine dimers by photoreactivation. Excision repair includes base excision repair, nucleotide excision repair, metal-directed mismatch repair, and translation DNA synthesis and the SOS response. In this video, we have covered the various organelles that make up a mammalian cell and their functions, followed by DNA replication, transcription, and translation. We also covered meiosis and mitosis, DNA mutation, and last but not least, DNA repair.
This video was done by Tricia! Wala!